All right, Sarah, go ahead and get started.
Great. Thank you, Meredith. So it is my pleasure today to introduce Professor Vickie Webster-Wood. Professor Webster-Wood received her PhD at Case Western Reserve University in 2017. She also completed a postdoc there as a Ruth L. Kirstein NRSA postdoctoral fellow, fancy names. So we were lucky to have her to join our department in 2018, where she has since established the CMU Biohybrid and Organic Robotics Group, the very creatively named B.O.R.G lab. And her research at CMU is focused on the use of organic materials as structures, sensors, actuators, and even controllers in robotics, as well as the development of many bio-inspired robotic and controller designs. She's received a slew of awards, including NSF Career Award, which is a big deal last year, and is co-PI of the NSF NeuroNext Network on Communication, Coordination, Control in Neuromechanical Systems. So we're very excited to hear about your recent work and look forward to being assimilated. So please take it away.
Oh, now you've stolen my joke.
Oh, no.
That's okay. Let me go ahead and get my screen shared. Awesome. Is that showing the correct screen?
Yes, you're on your title slide.
Wonderful. So thank you, Sarah. And hello, everyone. It's my pleasure to be with you all today to share some of the research that my lab, the Biohybrid and Organic Robotics Group, has been conducting over the past several years. And in particular, I want to talk with you today about our work in bio-inspired and biohybrid approaches towards creating lifelike and living robots. But before I get to the science, I really want to take a moment to acknowledge all of the great graduate and undergraduate researchers who have contributed to this work. Everything I'm going to show in the slides today is really the result of their hard work, and in particular, their adaptability in being able to, in many cases, completely change research trajectories in the face of the pandemic. So we're doing a lot of really cool things now that they have helped initialize because of all the changes we've had to make. So major kudos to this crew. Okay, so in motivating my work, I really love showing these videos of animals because animals are capable of tremendous feats of agility, strength, ingenuity, creativity. They adapt and respond to dynamic changes in their environment. They learn from experience, and they solve complex real-world problems. And as an engineer, these are capabilities that we want to capture in future robotic systems. However, unlike traditional robots, the biological systems that inhabit our natural world are already composed of dynamic responsive materials. For example, tissue growth is guided by biodegradable protein structures that can adapt and be remodeled by the cells. The muscles that allow us to move are adaptable and self-healing. And biological brains are capable of complex computation and learning. And with these materials in mind, in my lab, we really focus on bio-inspired and biohybrid robotics. We want to be able to learn fundamental principles of actuation and control by studying and modeling biological organisms, and ultimately even be able to harness the material capabilities that we see in these animals by identifying kind of renewable biocompatible materials and appropriate fabrication techniques that then let us create adaptable living actuators and energy efficient biological living controllers. And ultimately, our goal is to create soft bio-inspired robots that can themselves safely interact with us and our environment, with our hope being that one day these robots can actually be made of materials that are completely biodegradable and renewable. So today I'm going to share some characteristic examples of the work that we've been doing in these areas over the past 3 and about a half years, including some of our work in bio-inspired robotics for basic science, where we're developing neuromechanical models and robots as models for multifunctional behavior. Additionally, I'll share kind of 2 project areas we have related to biohybrid robotics, including our work on biomaterials and manufacturing, and some of the more recent work that we've been doing towards creating living robotic controllers. So let's start with bio-inspired robotics. In my lab, we see robots as not only tools for deployment in the real world, but also as controllable platforms for testing hypotheses in neuroscience and neuromechanics. And one of the key questions that really drives our research is, how do animals generate the multifunctional adaptable behavior that we see in the world around us? And this question is really at the heart of our NSF-funded Next Generation Network for Neuroscience Award. And more recently, we've actually spun out some of the basic science that we learned through this process and received some funding from the Foundational Research in Robotics program to adapt our robots for agricultural applications. And a number of students have contributed to this work over the years, as shown here. So to answer this question, using neuromechanical modeling and robotics, we really need to have a tractable animal model of adaptable behavior. For this, we turn to the sea slug Aplysia californica. So if you've ever wandered or down the B-level hall in Hammerschlag and wondered, why are Vickie's grad students messing around with aquariums in a tiny little closet in mechanical engineering? This is it. But in all seriousness, Aplysia is a robust marine sea slug. It has a relatively small nervous system, and in particular for the behaviors we study, the controlling ganglia really only have a couple thousand neurons total. Additionally, it's a model system in neuroscience, and so there's decades of research on the neuromechanics that we can leverage for our own research. As a mechanical engineer, Aplysia also has this really interesting, complex, soft body musculature. The animal is an invertebrate. It has no skeletal system. All of its structures are essentially muscular hydrostats or open circulatory hydrostatic skeletons. And it's really phenomenal when you look at these things that they can actually do anything at all. But perhaps most importantly for our research is that Aplysia demonstrates multifunctional behavior during feeding. And those behaviors include biting, which is an attempt by the animal to grab seaweed. And that's what you're seeing in this bottom right video. So if you've ever seen the movie Alien, it's kind of like that, where there's this internal grasping apparatus that reaches out between the jaws and grabs seaweed and pulls it back into the mouth. So you're seeing that inner grasping apparatus kind of come out between the jaws here. If the animal successfully grabs seaweed, it can then swallow that seaweed, and it can also reject or spit out inedible food. And it can transition between all of these behaviors and adapt the timing or forces of a behavior depending on external mechanical loads. And all of this is due to forces generated in this muscular hydrostat feeding apparatus that we call the buccal mass. The buccal mass is controlled by neurons in the buccal ganglia, which you can kind of think of as the motor controller. And that receives higher level descending commands from the cerebral ganglion. And we can break down the problem of multifunctional behavior into kind of two sections. First, we need to understand which neural units are needed to model our multifunctional neural controller. And second, we need to understand how the muscular itself may be contributing to multifunctional behavior through morphological computation. For our control model, our goal was initially to be able to predict and control multifunctional behavior in real time. And to achieve this, we need a tractable animal model, which we have in these sea slugs, and a computationally efficient model framework. So our solution was to create a hybrid Boolean continuous model of the feeding behavior in Aplysia. This is work that we're doing in collaboration with neuroscientists and mathematicians at Case Western Reserve University. And what we do is we start by looking for each behavior and seeing what are the key neurons and muscles that are involved on an individual basis. For example, we can start to study swallowing and try to figure out, how does the animal generate this multifunctional swallowing behavior? In doing so, we see that swallowing starts with kind of this pinch phase where the jaws are activated to close down on seaweed. This prevents the seaweed from being pushed out when the internal grasping apparatus is protracted forward. The grasper can then close again on seaweed and pull it back in through retraction. and the combined muscle activities result in a force on the piece of seaweed. Each of these boxed bursts of activity in the data from the actual animals represents the firing of individual named neurons that we can identify animal to animal. And we can approximate the activities shown here using Boolean bursts, where we represent the activity as either off, representing quiescence in the neuron, or on to represent firing. And so with this information in hand, based on our team's own prior work and the existing literature, we can build a model of the neural network in layers that takes into account the actual connectivity of neurons in the animal. First, we build this motor neuron layer based on those key neurons we saw in our animal recordings. Second, we add a layer of buccal interneurons. These interneurons kind of coordinate the activity of the motor neurons. And finally, we can add the cerebral interneurons, which are responsible for controlling behavioral switching in response to external sensory cues. We can then connect the motor neuron layer to a simplified biomechanical model of the feeding apparatus that provides sensory feedback back to the interneuron layers. We connected this with a really simple biomechanical model for our initial development, because we really wanted to be able to run the model faster than real time, to be able to do things in ensemble simulations and possibly even control the animal in real time, which we haven't gotten to yet. But what we needed to do was be able to model the quasi-static motion of the grasper and the head of the animal. So we approximated these as point bodies, which we connected to each other via springs to represent the surrounding connective tissue and musculature. And then we applied forces to the grasper or head as appropriate to represent each of the muscles. To best mimic the experiments that the biologists were doing, we simulate a piece of seaweed attached to a rigid force transducer. And we had this force threshold to allow the seaweed to break at different loads depending on the simulation we're running. And so the way that this kind of looks when we simulate it, just represented here in a schematic, is that we start at rest, the grasper is protracted forward and closes on the seaweed, and then the grasper is retracted back. But because we're simulating this kind of inedible seaweed, the head of the animal gets pulled up as it tries to retract. And this is the same thing that we see in the animal experiments. And then the grasper can release and the whole thing slips back to rest. And so when we combine these two models, we're able to qualitatively generate simulations of the actual animal behavior. And we can do that for biting by representing sensory cues that may induce the animal to bite, such as the absence of mechanical stimuli in the grasper, so the animal hasn't grabbed anything yet, but having stimuli at the lips. That's how we get the animals to eat in the aquariums. And we see that for these sensory signals, the grasper moves through a protraction-retraction cycle with the grasper closed. If instead we use external sensory cues that indicate that seaweed has been successfully grasped and is ready to be eaten, we see that the grasper has these really strong retraction phases with the grasper closed, and that generates a lot of force on the seaweed, just like we see in the animals. We can also have the simulation redirect material if we have a positive mechanical stimuli, but no chemical stimuli, which is what you might be seeing if the animal is trying to eat a piece of plastic tubing. And this is characterized by a strong protraction phase to push food or material back out that develops a lot of outward force. The model is also capable of behavioral switching. So if we simulate a sensory state change from a bite-like to a swallow-like state, we can get the grasper motion to kind of transition between the two behaviors. Perhaps most interestingly with this model, though, is the model adapts to changes in mechanical load as seen in the animal. So if we simulate a really weak seaweed, so seaweed that breaks at a really low force threshold, we see that the grasper moves through its protraction-retraction cycle, and that we start to develop force on the seaweed, but it breaks really quickly. And the motion of the grasper follows a more bite-like pattern. However, if we simulate strong seaweed, the force on the seaweed increases until the grasper releases, and then it drops back off. And this is exactly what we see in the animal. Whoop. Mouse problems. There we go. And why this is so exciting is because we didn't program this into our controller. This is an emergent property that comes from us having modeled the neural connectomics and musculature, at least as approximations of what's actually in the animal. And for those of you in the audience who care deeply about aplysia science, because I know there are so many of you, this is the first aplysia control model to include actual neural connectomics and a simulated model of the biomechanics with sensory feedback. So it was very exciting to be able to see these properties that we actually see in the animals captured in the simulation. Okay, so we've got this model. We can qualitatively predict the neural activity in the animal, but our initial neuromechanical model, we implemented a very simple approximation of the biomechanics. And we were just capturing the grasper and head motion as these 2-point bodies. While this was motivated by our initial goal for a computationally efficient controller, we do know that the mechanics of the system are quite a bit more complex. In fact, prior work using MRIs of the animals while they're swallowing shows that the grasper actively changes shape and rotates throughout feeding behavior. And this may vary the mechanical advantage of the surrounding muscles, thereby contributing to morphological computation. So towards understanding how this mechanical reconfiguration may influence control and behavior, we're creating a modular modeling framework in Simulink. This is work that's being done by Ashley Liao and Ravish Suknandan in my lab in collaboration with our biology friends at Case Western Reserve University. And our goal with this is to be able to use a demand-driven complexity approach to basically add more complex, more biomimetic models to our model and assess how that influences our ability to capture the actual animal behavior. So they started by implementing our original model, which just has a translational degree of freedom. And so in Simulink, they were able to reproduce the original model results, which is excellent. Ashley caught a bug in the code, which is excellent. And we ended up getting this all together into Simulink. And from there, they can start adding complexity. For example, we can add a rotational degree of freedom to try to capture that grasper rotation we see in the MRI. And they found that they were able to do this without losing multifunctionality, but that it did require a lot more model parameter tuning. Furthermore, we can start to look at how the actual models of our components influence our dynamics. We can replace the muscle models with more biomimetic muscle models. In our original model framework, we just used low pass filter approximations of smooth muscle. However, we can replace those with more complex models. For the I2, for example, there's a hill type muscle model in the literature that we were able to test. And in simulation, this had the largest impact on the model dynamics. So moving forward, we're now actually working on characterizing more of the muscles in the system so that we can add those to the model as well. And with each of these models, these are still relatively simple, and we neglect possible soft body interactions for the sake of computational speed. However, we know that these mechanics likely play a large role in behavior. Therefore, we're now creating a biomimetic robotic model of the aplysia buccal mass for testing these biological hypotheses of control and actuation with real-world physics. In our robotic model, we include physical representations of important feeding muscles and structures. This is work that a number of students are contributing to, being led by Kevin Dye, Michael Bennington, and Ravish Suknandan, with a great team of undergraduates who have been helping along the way. Okay, so for the robot, for the primary retractor muscle, I3, and the external I1 muscle, we use McKibben-style pneumatic artificial muscles. In order to achieve the larger contraction ratios that we needed to mimic aplysia muscle, these actuators are fabricated with a latex balloon as the internal bladder, housed within an overexpanded mesh. And then as the balloon inflates, the overexpanded mesh restricts its motion so that we get contraction along the axis of the actuator. The I1 muscle is relatively straight in the animal, so we use the kind of standard linear McKibben design. And for the I3 muscle, we approximated this as a series of toroidal rings. The I2 muscle, however, in the animal is a little different. It's very interesting. It's this broad, flat, Y-shaped muscle that is exceptionally thin. So to mimic the geometry of this, as well as the fiber orientations, what we needed to do was develop a very flat, broad pneumatic actuator. And for this, we took inspiration from work by Weirko et al. That they had created these little units that were these flat artificial muscles, where they had these kind of embedded fibers that allowed the contraction in response to inflation of these small chambers. And we recreated these in the lab and then worked on adapting this to our I2 muscle. In order to do that, we had to make some modifications from the original work reported in the literature. We created a Y-shaped mold and sequentially assembled and molded our actuator. Rather than using embedded Kevlar fibers for the kind of reinforcing fibers, we found that those kind of delaminated out, tore out pretty easily. So the guys had a great idea to use a Silhouette Portrait 2 electronic cutter and cut these polyester films in order to reinforce the actuator. And in doing that, they were able to kind of add these sequentially to this stacked mold design and then cast elastomer into the cavity as each layer is added. After curing the whole kind of stack, they're able to remove the mold and dissolve the PVA films with deionized water. And this gives us this flat I2 actuator. In order to mimic the internal grasping apparatus in Aplysia, a artificial odontophore was created using a 3D printed shell. And to mimic the soft grasping surface, we used a reinforced silicone that was adhered to moving jaws of the 3D printed shell. As we were building this robot, we recognized that the ability to detect successful grasping of the object or grasping pressure was going to be critical to our controller. And so we really needed some sort of force sensor that was soft and small form factor that we could embed in the surface. Fortunately, in this work, we were collaborating with Lou Lee and Dr. Howie Choset on another bio-inspired robotic project that we could leverage. So stepping away from Aplysia for a moment, Lou and Dr. Choset were very interested in whether we could give low-cost sensors the ability to aid in materials characterization for recycling. And so working with them, we took inspiration from human finger pads and created a magnet-based elastomeric tactile sensor. The sensor mimics the dermis and the epidermis of a finger pad, as well as allows you to cast these fingerprint-like ridges on the surface. And when the sensor comes into contact with an object, the elastomer deforms and the magnet moves relative to an embedded Hall effect sensor base. The sensors can be made in pretty small form factor and with different fingerprint patterns. And this work was funded by MFI, and a number of students, both from the B.O.R.G and Robotics Institute, have contributed to it. As part of this work, Kevin developed a fabrication pipeline to allow the sensors to be produced in bulk with these kind of multi-layer castings and magnet embedding. We also developed a computational model of the sensor to facilitate design and selected 3 designs for experimental testing on a number of different material surfaces. And so our goal here was to be able to extract frequency domain information from the sensor data and use that to classify which of the materials we were testing. So we tested a couple different fingerprint patterns, flat, just no ridges, straight ridges, curved ridges. And what we found is that the addition of these biomimetic features, these fingerprint ridges, really did improve our ability to classify the object surfaces. So that was really interesting and provided an interesting soft sensor for use in our Aplysia robot. So we combined this all together, and thereby our slug-like uniaxial grasper robot, or SlugBot, was created. And I take no credit for the name. That is all the Slug Squad's doing up there in the corner. Okay, so let's see this in action. To control SlugBot, we implemented our Boolean neural control circuit with minor modifications on a teensy microcontroller, and we stimulated external sensory cues using button inputs. And so here you're seeing SlugBot performing a bite-like feeding pattern. And of course, slugs are slow. So rather than watching the video, let's see how this stacks up relative to our prior model and the animal. So we tested the robot in both biting and swallowing, and we compared the robot's performance to our original model and found that they do produce qualitatively similar behaviors. Even more excitingly, we found that the robot's behavior is also similar to kinematic observations from those MRIs of animal swallowing. And while there's slight shifts in this translation profile here, this was a very exciting proof of concept that we can approximate aplasia mechanics with these synthetic actuators. It also gave us some good insight into some of the features that we need to update for version 2. Okay. However, there are several limitations with this sort of bio-inspired approach to robotics. For example, our synthetic actuators don't really yet reflect the actual properties of living muscle. They lack the ability to adapt in response to training, and they lack self-healing. For this work, our controller contains only a fraction of the neurons of the actual aplasia nervous system, and we didn't really capture biologically accurate neural dynamics by any stretch of the imagination. Additionally, it can't learn yet. We are currently continuing to work on this and developing bio-inspired approaches to address many of these limitations. For example, Ravish has led a project with Kevin in which we're developing some soft elastomeric magnetorheological dampers for inclusion in these actuators so that we can tune the force velocity properties to better mimic muscle. In this work, they'll be presenting at ICRA later this summer. We're also continuing to improve our controller. So I'm Zoom co-advising a PhD student at Case who is developing a synthetic nervous system controller of the aplasia feeding apparatus to give us a little bit more biologically realistic neural dynamics. And so we can continue working on the kind of these bio-inspired approaches, but we also kind of have this question in my lab of how can we leverage the material properties of living materials directly in our robotic systems? And this question's at the heart of my NSF-funded career award, as well as 2 kind of recently awarded proposals being led by Dr. Jessica Zhang that we're collaborating on from PETA and PMFI. And again, a number of students have contributed to this work. Ultimately, in addressing this question, my long-term goal is to create completely adaptable, biodegradable robots. Okay. So let's start out by looking at some of the work that we've done in trying to identify materials for use in these robots and create fabrication techniques for using with those materials. To achieve our long-term goal, we really need active, adaptable robotic materials that are biocompatible, renewable, and even biodegradable. We think this is particularly important for applications in biointerfacing and medicine, or environmental deployment in sensitive ecosystems, where in both environments, your robot really needs to be safe for operation in that environment. One material that shows a lot of great promise for biohybrid robotics actually comes from prior literature in tendon regeneration. And this is electrocompacted and aligned collagen. Using collagen, like a viscous molecular collagen solution, we can actually apply a voltage across the solution, and the collagen molecules become charged and compact along the isoelectric point. In doing this, we can create really robust threads and sheets composed entirely of collagen. For those of you not in the biomaterial space, collagen is an extracellular matrix protein that muscle cells really like to attach to. And we can use the collagen to align muscle that we grow on top of it with the degree of alignment depending on the alignment of the underlying material. During my own PhD, I demonstrated that we could fabricate robot bodies using this approach kind of en masse, and that they did indeed result in alignment of muscle cells on these substrates. Using this approach, the robots even moved in response to electrical stimulation. Many of you have seen this video. I promise it is moving. The robots back then, I'm not going to say how long ago back then, I guess it's on the slide, oops, were very slow. And there's a couple of things that we can observe from watching these robots. One is that we got really limited deflection of the legs. So we had a really low stride length. We were also getting really low muscle force. And so we wanted to try to address these issues. When I first started here at CMU in 2018, we began to look at this problem again. And we asked ourselves whether we could create ELAC threads and sheets that had material properties more amenable to muscle culture. And this led to the question of how do ELAC material properties vary with manufacturing parameters? There are a lot of knobs that we can tune in the production of these materials, including how long we apply a current, what voltage we use, and what the collagen concentration is. And so my PhD student Wenhun Sun designed a series of fractional factorial experiments to kind of investigate the individual effects of each of these parameters, as well as any joint interactions that we might see. For the full study results, I encourage you to read our paper in the Journal of Biomechanical Engineering, but some of the key takeaways include that ELAC fibers made with longer isoelectric focusing duration showed significantly higher tensile strength, failure strain, and a larger cross-sectional area. And ELAC fibers made with lower isoelectric voltages exhibited significantly lower tensile strength, modulus, and a smaller cross-sectional area. We also see kind of joint effects from a number of these different manufacturing parameters. And what this does is it allows us to really tune the mechanical properties of this material across quite a wide range of tensile modulus, tensile strength, and diameter. And Wen actually also developed a deep learning based recommendation tool that he trained on this data set in order for a user to be able to provide their desired properties of the resulting thread and have the tool tell them what manufacturing parameters they should use. So while the ability to tune the substrate properties should help with the limited deflection that we saw in our robots, we still have fairly immature muscle tissue. This is a known phenomenon in tissue engineering, where the 2D cultures kind of fail to capture the actual native environment that we see in vivo. And so 3D tissue culture is one way to improve tissue performance and maturation. As a result, recent years have seen biohybrid robots move away from the thin film monolayer cultures that you see in early robots to 3D tissues. These are typically formed via casting or more recently via bioprinting. However, you may notice from these results that we see in the literature that these are all relatively small scale, and that the muscle is attached to very soft or very lightweight structures. This is in part because of part of the research that somehow seems to never end up in the papers, right? And that is high failure rates, possibly due to stress concentrations between very soft muscle tissues and any stiffer robotic structures. The muscle has a very low modulus. And so you can see that when we try to attach it to kind of more rigid structures, that can increase the likelihood of getting these tears in the tissue. Well, we looked at this and we said, hey, we've got this really great biomaterial. We know how to tune its mechanical properties, and we know that the muscle loves to attach to it. Can we combine this with 3D muscle culture to create bioactuators with tendon-like ends so that these bioactuators could be used on a broader range of robotic applications? And this is work that we're doing in collaboration with Dr. Adam Feinberg in BME to integrate fiber reinforcement into the freeform reversible embedding of suspended hydrogels, or fresh prints process. You gotta love the puns. This is a technique that his group developed to allow soft hydrogel and biomaterials to be printed into a support bath so that you can print overhangs and very small features that would not otherwise be achievable. The process is compatible with a number of different bioinks and hydrogels that are of interest to the tissue engineering community. And so this is a really great fabrication platform for these types of approaches. However, the resulting printed structures and materials are still very soft and fragile, right? Just like 3D muscle culture. To overcome this limitation, we developed a new fiber reinforcing printhead that allows prefabricated fibers to be embedded in the hydrogel during printing. We combine this with a Replistrator syringe pump and an open source 3D printer in order to create a multi-material printing platform. The long fiber extruder is composed of 2 guiding tubes to direct the fiber through a pair of soft silicone driving wheels to allow the fiber to be extruded without damaging the fiber. In our LFE fresh process, or long fiber embedding fresh process, we begin by printing the primary hydrogel into the support bath. And then fibers can be embedded either kind of layer by layer or embedded directly in 3D within the hydrogel. And we can go back and print additional hydrogel as needed to encapsulate the fibers. After we're done printing, we can then liquefy the support bath for part retrieval. Of course, as this process is essentially trying to push rope into jelly, the printability of any given fiber is going to depend on the fiber properties and the gel properties. If the gel is too stiff or the fiber is too compliant, then the fiber is going to buckle. So we developed both analytical and computational models of the printing process to allow us to provide a printability model for the process. And we validated this experimentally across a number of fiber types and sizes. When fibers within that green printability region are printed, we can fully encapsulate them within the hydrogel. And we see sub-millimeter deviations from our desired print paths in our initial prints. More recently, Wen has also demonstrated that we can improve, we can improve print fidelity using a closed loop computer vision based feedback control process. For applications where robust thread gel adhesion is needed, we also tested for this interaction through a series of pull-out tests. We found that the initial pull-out force required was pretty low, but that we could increase adhesion strength through cross-linking. Furthermore, one of the primary benefits that we anticipated with LFE Fresh was the ability to structurally reinforce these printed hydrogels. To test this reinforcement, we performed tensile testing with and without fiber reinforcement. And we found that, as expected, LFE Fresh printing significantly increased the ultimate tensile strength of our composites. As one would expect, this reinforcement also improves composite strength in bending. LFE Fresh is compatible with a wide range of threads and hydrogels. Wen has printed nylon, polyester, I think silk, collagen, very tiny wires, some strange threads that he made up himself. He's printed them in alginate and collagen. And we can use this process to print single threads or multiple threads in a single path, both in 2D or in 3D. And so moving forward, we're now integrating this printing platform into 3D muscle tissue printing in order to embed elac tendons in muscle tissues during manufacturing. And the crew is also developing some really interesting culture vessels that will allow us to really dynamically monitor these tissues in real time in work that Saul Schaefer's been leading. Okay, now, I know I don't have much time left, but I also wanted to share with you some of the work that we're doing towards biohybrid control. While you already saw that we have developed these bio-inspired control models in silico, there are limitations in translating that to biohybrid robotics. For example, during my PhD, we found that stimulation via natural neural patterns significantly increased muscle force and longevity over electrical stimulation, either to a nerve or in a surrounding bath of media. Therefore, my team is really interested in the possibility of growing our own biological neural controllers that we can interface with our bioactuators. But because we're engineers, we don't want to just throw a bunch of neurons in a dish and call it a controller. We want to be able to design the structure of the controller, predict the behavior, and ultimately kind of train or reprogram those biological neurons for the robotic tasks that we want them to perform. And so this is work that we're doing in collaboration with Dr. Jessica Zhang. It's being led by our two co-advised PhD students, Ashley Liao and Quanren Qian. And we want to develop these predictive models of how our neurons are going to grow so that we can do controller design. However, to do that, we need to understand neuron growth in vitro. From looking at the literature, we found that neuron growth is typically talked about qualitatively in terms of growth stages. And while that's great in biology, it's really hard to then have metrics to determine whether your models are doing a good job of predicting the neuron growth that we're seeing. So we wanted to start by identifying quantitative metrics of neuron growth that we could use for validating our computational model or tuning our computational models. To do this, we cultured a whole bunch of rat hippocampal neurons and just on unpatterned substrates. And Ashley and her team of undergrads used semi-automated tracing techniques to trace the individual neurites on hundreds of cells. Based on that, Ashley then really worked to identify quantitative metrics that could be used to distinguish between different neuron growth stages. We started by looking at some common morphometrics that had previously been reported in the literature for telling different cell types apart. These included degree, which is kind of the number of these neurite tips, total length, and tortuosity. And from our data set, what we found is that the total length and degree varied significantly across the growth stages that we studied. Additionally, we ultimately want to be able to tell whether the direction of growth is being controlled by external cues, right? If our goal is to pattern these controllers, we need to know if the pattern is actually guiding them. So we needed a new metric to capture that kind of goal-directed growth. For this, we took inspiration from the change point test that was originally introduced by Byrne et al. To quantitatively describe animal wandering. And using this test, we can break down these wandering neuron paths into a number of segments that are separated by points at which there's a significant reorientation of the path heading. And from this, we get two new metrics, kind of the number of change points and the average segment length. For our data set, we found that both the average segment length and number of change points varied significantly between our time points. These results gave us quantitative metrics that we could use for validating neuron growth models. And as part of this collaboration, Quanren has been developing a phase field model of neuron growth to actually simulate these growth processes in silico. And in our first publication on this work that has now gotten past the first round of revisions and is back out for review, we show that this model can be used and tuned to reproduce some of the actual neuron structures that we see in our data set. And so next up, we'll be adapting both our culture and modeling approaches to pattern these neural circuits and create biological neural controllers. So far in these slides, we've looked at a lot of ways that we're studying the biology and trying to predict the biology and understand the biology. But I also wanted to share how studying these types of biological phenomenon can be used to bring information back into engineering. For example, we have an ongoing project in collaboration with Dr. Amir Barati-Farahmani, in which Kevin Dye has been working on developing techniques to grow the connectivity of artificial neural networks. And where this work really came from is when we look at artificial neural networks, we often see that they have these structures that are highly connected, right? Often even fully connected. And of course, this is at odds with the actual connectivity in biological brains. In our brains, we see that our networks are really sparsely connected. And this may be due to computational efficiency. This may have some effect on how our brains are actually able to process all of these complex things. And we wanted to see if we used kind of inspired, abstracted models of neuron growth, could we grow connectivity in these networks and improve performance on machine learning tasks? And so Kevin developed two approaches here, a stochastic growth approach, where we start by initializing all of our neurons in kind of this spatial growth map, and then growing axons, quote unquote, from input layer neurons to hidden layer neurons, where we define random growth angles and growth rates. And he also developed an approach to more closely mimic the environment in biological brains by introducing chemicals being released from hidden layer neurons and creating a gradient in our growth map. In response to this gradient, the growth angle and growth rate could then be adapted and changed. In both approaches, once a connection has reached a certain radius around the hidden layer neuron, we form the connection, and the number of connections between any two given nodes sets the initial weight. And so Kevin grew just a whole bunch of these networks in simulation and tested them on the MNIST benchmarking data set. And so MNIST is a whole bunch of handwritten numbers, and the goal is to essentially show the neural network a picture of one of these handwritten numbers and have it output the correct digit. And what's really interesting is that Kevin found that for even really relatively low connectivity percentages, so very sparse networks, both of our growth-based approaches significantly improved our classification accuracy over a fully connected network. And so this work is ongoing, and we're working on a manuscript in preparation on this. Okay, so I think I'm coming to the end of my time. I need a clock in this room, but I wanted to wrap up with some future work, right? We've got a lot of kind of foundations established in a number of these different trajectories, but we see a lot of really exciting ongoing challenges that we can address over the coming years. For example, in terms of the biomaterials we're working with, we're starting to move towards more complex robots where we really want to be able to get these outside of these petri dishes they currently have to live in. And so we want to be able to add packaging and life support, as well as scalable manufacturing techniques so that we can move away from the grad student with tweezers method. We need to develop techniques for our actuators to improve their strength capabilities, or to bring more biomimetic mechanics into our synthetic actuators. And as we're starting to grow these patterned neural controllers, we really want to be able to train them and program them to have the biomimetic behavioral flexibility that we see in animals, with all of this being towards our long-term goal of autonomous biodegradable robots. I'd be remiss if I didn't take another moment to acknowledge all of the people who make this science possible, including my research lab and our collaborators both here at CMU and around the world. The work has been funded by the NSF through a Next Generation Network for Neuroscience Award, a Career Award, and our recent Foundational Research in Robotics Award. Like I said, we've had more local funding from MFI, a Berkman Faculty Development Fund, PETA, and PMFI. And the crew has also been fantastic at getting their own funding. And we're supported by 3 graduate research fellowships and 6 CMU summer undergraduate research fellowships.